Dear students, today we revise the male reproductive structures in flowering plants. The male reproductive structure are stamens, and the stamen consists of two parts: the filament, which is a long and slender stalk-like structure, and the terminal part is known as anther. The proximal end of stamens is attached to the thalamus or the petals. If we observe the stamens with the help of dissecting microscope, then we observe that the length of filament and the shape of anthers are vary in different species, in different flowers of different species. The number of stamens are also different in different flowers of different species. The anther is a bilobed structure, and each lobe having two theca means dithecous. The anther is a tetragonal structure and has 4 microsporangia at each corner, and later these microsporangia develop into pollen sacs. Now we study the structure of microsporangium. Each anther has 4 microsporangium at its corner and surrounded by different types of tissues, such as the outermost layer is called as epidermis, and below epidermis the endothecium is present, and inner to the endothecium 2 to 3 layered middle layer is present, and the innermost layer is called as tapetum. The outer three wall act as protective layer, and the innermost layer, the tapetum, nourishes to the developing pollen grain. Each microsporangium has a homogeneous tissue or cells which are known as sporogenous tissue, and the cells of this sporogenous tissue is capable to produce a microspore tetrad. It means each cell of the sporogenous tissue is a pollen potential or the pollen mother cells, and these pollen mother cells or the microspore mother cell undergo the meiotic division to form a microspore tetrad. Each cell of microspore tetrad is haploid, and as the anther matures, and dehydrates, these microspores dissociate and later develop into pollen grains. And each microsporangium has several thousands of pollen grains. And as the anther dices, these pollen grains are released. Now we study the pollen grains. The pollen grains represent the male gametophyte, and each microspore or pollen grain has a spherical structure which is surrounded by two layers. The outer layer is called as axine, which is formed of hard substances known as sporogenous, known as sporopollenin, and this layer has some pore-like structures known as germ pores, where the sporopollenin is absent, and the inner layer is called as entine, which is a continuous layer and formed of cellulose and pectin. The cytoplasm of the cell is surrounded by a plasma membrane, and when the pollen matures, it has two-celled structure. The large cell is called as vegetative cells, whose nucleus is irregular and large, whereas the other cell is smaller cells and floating in the cytoplasm of the vegetative cells. And in the vegetative cells divide to form, the generative cell divide to form two male gametophyte, and in this way the pollen grain has three celled stage. About 60% species, the pollen grain released in the two-celled stage, whereas the remaining rest plants release the pollen grain at three-celled stage. Thank you.